For more than a century, the name alone told everyone what they thought they needed to know. Saber-toothed tiger, a bigger, older, more savage relative of the striped cats prowling Asia today. That assumption survived fossils, survived museum placards, and even survived science's first real attempt to test it with actual genetic evidence. When researchers finally sequenced Smilodon's DNA in the early 1990s, the results seemed to confirm the name outright, placing the animal squarely inside the modern cat family alongside lions, tigers, and house cats. It took another 25 years, 3 separate research teams, and one deeply embarrassing contamination error to find out that first answer was completely wrong from the start. Start with what Smilodon actually was, because almost none of it matches a tiger in the way the name suggests. Built with disproportionately massive forelimbs designed for grappling and pinning prey at close range rather than chasing anything down over open ground, Smilodon hunted nothing like a modern big cat does. Its jaw could open to nearly 120 degrees, almost double what a modern lion can manage, driving 2 canine teeth up to 7 inches long into the throat of prey, many times its own body weight in a single precise strike rather than a prolonged, drawn-out struggle. The genus was formally named in 1842 by Danish naturalist Peter Lund, based on fossils recovered from limestone caves in Brazil that Lund initially mistook for the remains of a hyena before recognizing the distinctly cat-like skull structure underneath the surface. The North American species, Smilodon fetalis, followed in 1869, described separately by paleontologist Joseph Leidy from a single jaw fragment found in Texas. For roughly 2.5 million years, Smilodon ranged from what is now Canada down to the southern tip of South America, one of the most successful large predators the continent had ever produced before disappearing entirely around 10,000 years ago alongside most of the rest of the America's large mammals. That disappearance left behind one of the richest fossil collections of any prehistoric animal on Earth, and for well over a century, paleontologists had only that physical record to work with, no matter how many bones piled up in museum drawers around the world. Bones alone can only say so much about how closely two species are actually related on a genetic level. A separate group of extinct predators, nicknamed the false saber-tooths, evolved teeth and skulls superficially similar to Smilodons, despite belonging to an entirely different, unrelated family entirely, and telling the two groups apart, let alone figuring out exactly where Smilodon fit relative to living cats, required a level of precision skeletons simply couldn't provide on their own, no matter how carefully they were measured. The question sat unresolved for generations. Was this animal a true, if extinct, member of the same family that includes today's lions, tigers, and house cats, or something that only looked the part on the surface while actually belonging somewhere else entirely on the tree of life? If you're finding this interesting, be sure to subscribe to understand more DNA from the past. Getting a real answer meant reading actual genetic material, and Smilodon offered a genuine opportunity to try where many other extinct animals didn't. Genetic material has a hard ceiling of roughly 1 to 1.5 million years under ideal preservation conditions, and Smilodon had only been extinct for about 10,000, comfortably inside that window with plenty of room to spare. That timing mattered enormously. Animals like dinosaurs, extinct for tens of millions of years, sit so far outside that preservation window that recovering their actual DNA is considered essentially impossible by any known method, leaving researchers to rely entirely on bone structure and, at best, degraded protein fragments. Smilodon sat in a much more promising position, recently extinct enough that its genetic material should theoretically still exist somewhere in well-preserved remains, if researchers could just find a specimen good enough and a technique careful enough to read it accurately. In 1992, a research team led by geneticist Stephen O'Brien published a study in the Proceedings of the National Academy of Sciences describing a genuine technical breakthrough, a method for extracting usable genetic material from 14,000-year-old Smilodon fatalis bones preserved in asphalt at Rancho, Brea, the tar pit site in Los Angeles that has produced more Smilodon fossils than anywhere else on the planet by a wide margin. Extracting any usable DNA from bone this old was itself a significant achievement at the time, years before ancient DNA research had developed anything resembling the strict contamination controls considered standard practice today. The team sequenced 2 gene segments, one mitochondrial and one nuclear, from 3 separate fossil specimens, then compared those sequences against 15 living carnivore species spanning both cats and other, more distantly related carnivore families used as an outside comparison point. Their conclusion placed Smilodon inside the modern radiation of cats, closely related to the living lineage that eventually produced lions, tigers, and house cats, and clearly distinct from the false saber-tooths that had confused paleontologists for so long. For a molecular study of an extinct predator this significant, in an era when ancient DNA research was still brand new and largely untested, the finding was treated as a genuine advance. It shaped how the animal's ancestry was discussed for years afterward, appearing in textbooks and documentaries as settled science that nobody thought to seriously question again. It wasn't settled at all, and the confidence surrounding that original finding turned out to be entirely misplaced. In 2005, a separate research team, publishing in the journal Current Biology, used different fossil material and entirely different techniques and arrived at the opposite conclusion. Smilodon belonged to a lineage entirely separate from living cats, not nested within it the way the earlier study had confidently claimed to the wider scientific community. That contradiction alone was enough to cast serious doubt on the 1992 result, but it didn't yet explain how 2 teams working on the same basic question, using genuinely careful methods on both sides, could land on such completely different answers. A follow-up study the next year found the likely cause, and it wasn't a flaw in the math or the sequencing technology itself. The original nuclear DNA sample had almost certainly been contaminated at some point during handling or laboratory amplification by genetic material from an ordinary domestic house cat, the kind of animal that could easily have been present in or near the same laboratory space. Contamination detection in the early 1990s was nowhere near the rigorous standard it is today, with none of the dedicated clean rooms, negative controls, or statistical flagging methods that ancient DNA labs now consider absolutely mandatory before publishing any result of this kind. A handful of stray cat cells, introduced at any point between field collection in the tar pits and final amplification at the lab bench, were apparently enough to steer an entire phylogenetic conclusion in the wrong direction for more than a decade of published, widely cited science. The animal researchers had unknowingly compared Smilodon to, in the study that first placed it among modern cats, may have quite literally been sitting in the same building the whole time the experiment was running, an ordinary house pet accidentally rewriting the ancestry of an extinct predator 20 million years removed from it. Contamination errors like this one weren't unique to Smilodon, and the field's response over the following decades reshaped how ancient DNA research operates entirely. A fossil sample, however carefully excavated, spends its entire existence surrounded by modern organisms, and modern DNA is vastly more abundant and far easier to amplify than the degraded, fragmented traces locked inside old bone. Researchers eventually developed dedicated clean room facilities physically separated from any modern DNA source. Negative controls run alongside every sample to catch contamination before it reaches a published conclusion, and statistical methods capable of flagging results that look suspiciously modern rather than authentically ancient. The Smilodon correction stands today as one of the clearest before and after case studies in the field for exactly why those safeguards became standard practice rather than an optional precaution nobody bothered enforcing. Subscribe to not miss the next one. The correction didn't stop there, and the field wasn't finished tightening the picture. In 2017, a team led by geneticist Johanna Pijmans, again publishing in Current Biology, reconstructed complete mitochondrial genomes from Smilodon populator, the larger South American species, alongside 3 specimens of Homotherium, A separate genus of saber-toothed cat, known as the scimitar cat for its shorter, more finely serrated canines, distinct from Smilodon's longer, blade-like teeth, built for a completely different kind of killing bite. The results were unambiguous. Smilodon and Homotherium had diverged from each other roughly 18 million years ago, long before either lineage looked anything like the animal that would eventually go extinct, and both lineages shared a common ancestor with every living cat species alive today, around 20 million years ago. Pagemans put the scale of that divergence in blunt, easy-to-grasp terms afterward. In terms of mitochondrial DNA, these two extinct saber-toothed cats are more distantly related to each other than a tiger is to a modern house cat sitting on someone's couch right now. The 1992 study hadn't just placed Smilodon slightly closer to living cats than it should have been. It had taken an animal separated from modern felines by 20 million years of independent evolution and dropped it directly inside their family tree as if it belonged there all along. Final confirmation came when researchers sequenced the first draft nuclear genome ever recovered from Smilodon populator, using a specimen roughly 13,180 years old, discovered in Chile. The oldest complete paleogenome ever recovered anywhere in South America, a continent where hot, humid, chemically unstable soil typically destroys ancient genetic material long before anyone gets the chance to study it properly, making most South American fossils genetically unreadable no matter how well the bones themselves survive. The nuclear genome confirmed the same 20 million year divergence already found in the mitochondrial data, then went a step further, directly testing for gene flow between Smilodon and the lineage leading to living cats, the same kind of interbreeding that has been well documented among several modern big cat species elsewhere in the family. They found none, not a single trace, anywhere in the genome, across 20 million years of separation and coexistence on the same continent. That same study also solved a separate, smaller mystery that had puzzled researchers for years. Despite Rancho Brea holding more than 130,000 individual Smilodon bones, representing over 2,000 separate animals, the single largest collection of this species found anywhere on Earth, researchers concluded that recovering reliable ancient DNA from these specific asphalt-preserved specimens simply isn't feasible using any current method available to science. The tar that preserved the bones so beautifully for tens of thousands of years, protecting delicate skeletal detail in remarkable condition, turned out to be chemically hostile to the genetic material locked inside them, breaking the DNA down even while it protected the bone itself from decay. The breakthrough genome had to come from an ordinary skeletal specimen in Chile instead, thousands of miles from the largest saber-toothed cat graveyard on Earth, because that famous graveyard turned out to be one of the worst possible places on the planet to actually read its DNA. Line up all four studies, and the picture that emerges is a complete rewrite of where this animal actually belongs on the tree of life. It was never a big cat that happened to grow oversized canines, and it was never the tiger its own common name still implies to anyone hearing it for the first time. It was the last surviving member of an entirely separate branch of the cat family tree, one that split away before lions, tigers, and house cats had even begun to differentiate from each other, and one that never once exchanged genetic material with that branch across 20 million years of coexistence on the same planet. Its massive forelimbs, its extreme gape, its blade-like canines built for a precision throat bite rather than a crushing grip, evolved independently, arriving at a superficially cat-like predator through convergent evolution rather than close kinship. The same evolutionary pressure producing a similar-looking solution in two lineages that otherwise share almost nothing in their recent ancestry. What actually killed Smilodon off around 10,000 years ago remains less settled than what it actually was, with researchers still weighing some combination of a warming climate, declining prey populations, and new human hunting pressure rather than pointing confidently to any single confirmed cause the way the identity question now has one. But the identity question itself, the one the animal's own name got wrong from the very beginning back in 1842, took 25 years, 4 separate research teams, and a fossil from the one continent notorious for destroying the evidence scientists needed most to finally close for good. It was never a tiger. It never even came close, and the DNA that finally proved it had been waiting inside a Chilean fossil the entire time, thousands of miles from where anyone had originally thought to look.